Hello, welcome to OHBM. My name is Boli. I am from the Erasmus Medical Center. Today, I will introduce our work on the high-resolution autoencoder for construction of interpretable brain MRI endophenotypes. As the title of the abstract says, this talk will be about the deep encoding and decoding of brain MRI imaging. Specifically, it's about why do we need dimensionality reduction? How can we encode the data into low-dimensional endophenotypes? And finally, from methods to applications, what can we do with endophenotypes? And what can be decoded from them? In population neuroimaging studies, it is commonly a phenotype of interest rather than the raw image voxels that is used for the analysis, because of the high-dimensional nature of imaging data versus the relatively small number of the observations, namely the number of subjects. However, selecting the suboptimal phenotype, such as a specific brain region, can lead to misleading results or the loss of statistical power, as the underlying etiologies can have far more complex patterns beyond our anatomical knowledge. To eliminate the need and the bias of phenotype selection, unsupervised dimensionality reduction techniques can be applied to construct compact representations, namely the endophenotypes. Here we propose efficient and accurate deep learning algorithm for interpretable dimensionality reduction of high-resolution neuroimaging data. Within the category of deep learning, what makes one algorithm differ from another are generally the neural network architectures and the objective function. Autoencoder is a type of deep learning approach for unsupervised dimensionality reduction. It takes one image as input, encodes it into a compressed low-dimensional feature, and tries to reconstruct the same image by decoding the features. By maximizing the similarity between the input and the reconstructed image, we assume that the compressed features can sufficiently represent the input image. Namely, the essential information of the images are well preserved. Despite the promising performance of autoencoder in computer vision field, for instance, to analyze a picture of the digit, it is, however, non-trivial to apply such a method to neuroimaging data. The state-of-the-art autoencoders for medical imaging are currently limited to 3D regions of interest or 2D slices only. To solve this problem, a new technique has been developed to train on 2D slices and output a 3D image. Unfortunately, this approach cannot converge at high resolution. This result, however, is valuable, as it suggests that the GPU memory might not be the bottleneck for this task. So what is the source problem here? We expect that the challenges underlying the iceberg can be the complex 3D pattern of human brain and also the nonlinear spatial relationship across a dataset. To solve these challenges, here we propose a new approach for dimensionality reduction of high-resolution neuroimaging data. Recap what I have mentioned earlier, that the neural network architectures and the objective functions are the key components of a deep learning algorithm. Sometimes a good preprocessing strategy can also make a large difference than simply increasing the complexity of the network architecture. For the neural network architecture, we empirically optimize it for brain MRI analysis and for good learning ability with affordable amount of trainable parameters. The objective function, which is also known as the loss function, is the driving force of the deep learning car, because the training procedure is basically to estimate a set of parameters that can optimally minimize the value of the loss function. To encourage the learning of both intensity and the structural similarity, both global and local features, we propose a joint loss function that measures the correspondence between the input and the reconstructed images based on the smooth mean absolute error and the normalized cross correlation. Last but not least is pre-processing. As we can see here, there are not only anatomical variation, but also spatial variation across a neuroimaging dataset, which in itself is a challenging task called image registration. Therefore, to ease the learning procedure for anatomical features, we apply spatial normalization as a pre-processing step. We compute the modulated gray matter maps using voxel-based morphometry that describe the local gray matter density. From the population-based Rotterdam study, we included 11,000 3D T1-weighted brain MRI scans. After preprocessing, the input images are in the MNI standard space, 1 mm isotropic and with background crop. Now let's move on to the experiment and the result section. First, we evaluate the reconstruction performance of the trained autoencoder on 1,000 test images. The accuracy of reconstruction was measured by the Pearson's correlations between the intensity of the input and the reconstructed images across 78 brain regions on the Hammers atlas. The average Pearson's correlation over the regions was 0.77. For instance, this figure here shows a correlation for the right hippocampus, for which the Pearson's correlation was 0.71. Secondly, in two proof-of-principle experiments, we evaluated the applicability of the derived endophenotypes, namely the Zin brains, to downstream tasks, to predict age using linear regression, and to predict sex using logistic regression. The test accuracy for age prediction is measured by root mean squared error. The method yielded a root mean squared error of 5.14 years, lower than that reported in the previous work of 6.9 years. The bottom figure here shows the reconstructed age-specific image, where the relationship between the endophenotypes and the ages, as well as shows the revealed brain changes over aging, in which, for instance, the regions in red can be considered as brain atrophy. The test accuracy for sex prediction is measured by the error under the curve. The method achieved an AUC of 0.99, showing a high discriminative power of the endophenotypes. Similarly, we provide a semantic interpretation for sex-specific modulated gray matter maps. And in the colored image, the sex-relevant brain changes across male and female. With the above content, I would like to conclude that to the best of our knowledge, we developed a first high-resolution 3D autoencoder for brain MRI analysis, which is able to encode essential information of the image and to derive interpretable neuroimaging endophenotypes. As proof of principle, we associated the derived endophenotypes with age and sex, and achieve the state-of-the-art accuracy, efficiency, and interpretability at high resolution. We are currently working on analyzing more target variables, and we expect that such a method can be widely used in population neuroimaging studies. Also, we expect that the derived endophenotypes can serve as a valid biomarker for disease diagnosis and prognosis. With my last slides, I would like to thank all my co-authors, and I would be very happy to take your questions and discussions. Thank you.